Good morning, good afternoon, and good evening, everyone, from wherever you're joining us, and welcome to today's webinar titled Peripheral and Cerebral Vascular Responses Following High-Intensity Interval Exercise, featuring Dr. Bert Bond, Senior Lecturer, and Max Weston, PhD Candidate and Associate Lecturer, both within Sport and Health Sciences at the University of Exeter. Today, they will present an overview on their study investigating the effects high-intensity interval exercise has on cerebrovascular health. I wanted to take this moment to thank our partners at the American Physiological Society and the sponsor for this event, AD Instruments, for making it all possible. AD Instruments creates industry-leading software and hardware tools for life science researchers and educators. For over 30 years, AD Instruments' mission has been to support scientific discovery, and they are proud to be in strategic partnership with the American Physiological Society to support innovation in the life sciences. AD Instruments offers a range of solutions for accurate physiological measurements, and with the ability to integrate multiple data sources, systems can evolve as your experiments do, ensuring quality results wherever your research takes you. I'm ready to welcome our speakers, Bert and Max, to join me. Hi guys, thank you for being here today. Please take it away whenever you're ready.
Okay. Thanks, Sydney, and thank you everyone for joining us wherever in the world you are. Just wanted to start off by thanking as well the American Physiological Society, obviously, for supporting this talk, and AD Instruments for supporting the work that we do in our lab. You can't perform this type of research without really good technical support. So, huge thanks to AD Instruments. And yeah, Sydney as well, thank you to you for helping organize this and dealing with academics in different time zones. So that was much appreciated. Thank you. I've made all these slides available, and I just draw your attention to our email addresses are on this screen in front of you here. So if there's anything that we perhaps gloss over too quickly or a bit more information that you'd like, feel free to either email myself or Max, and we're quite happy to open up lines of communication with you and talk a little bit, perhaps a bit more in detail about some of the things we're going to discuss here. But the function of this session is really, again, to talk about some of the vascular work that we've been doing here at the University of Exeter, and particularly focusing on one of our publications in Journal of Applied Physiology, which included some of these vascular techniques. So we're going to kind of use that as the basis of much of the things we talk about today. So just by way of a quick introduction, I'm a senior lecturer in public health and sports science, which is part of the University of Exeter Medical School here at Exeter. And my research interests focus around lifestyle risk factors for cardiovascular disease. And this sort of typically focuses around performing mostly interventional studies and considering vascular function as a key research outcome for that work. So when I talk about lifestyle risk factors, I'm really thinking about things that we do to ourselves. So this includes physical activity or exercise. The two aren't the same. Similarly, diet and nutrition, concussion and head impacts. We're doing a lot of work in that space. And then a recent development is trying to consider sleep or lack of sleep on certainly cerebrovascular function. But we're trying to understand how these things might relate to cardiovascular disease risk by trying to understand what's happening to vascular function. Max, you just want to say a quick hello?
Yeah. So hello, everybody, and welcome from myself as well. So I'm currently working as an associate lecturer in the same department with Bert. But Bert's also my PhD supervisor. I'm writing up for completion this summer. And my research predominantly around exercise physiology and the cerebrovascular responses to exercise, looking at two main independent variables. So the effect of exercise intensity on these responses and the effect of age and maturation, particularly in the development from childhood through to adulthood. And we'll be focusing this talk on a particular study we did in adults.
Thanks, Max. Okay, so again, just for those not in the know, the University of Exeter is in Devon, with the beautiful county of Devon in the southwest of England. This is where we're based, and we're actually, our lab, our group is based on St. Luke's campus, which is a very small kind of campus in Exeter. Exeter has many different campuses, and everyone walking around St. Luke's campus is either studying medicine, sports science, or teacher training, give or take. So we're surrounded by sort of like-minded individuals. And the University of Exeter is a Russell Group institution, which means that we are, profoundly research active. And in fact, sports science or research at sports science returned as number one in the Research Excellent Framework in 2021. So it's the latest research assessment exercise we have in the UK, which ranks kind of research by different UK institutions. So we're really research active. And I mention this just so you know where we are and kind of what we do. And if you're looking to collaborate, or if you have students in your lab that are looking to spend some time abroad, perhaps, or in a different institution, then we've got quite a liberal open door policy. We're quite keen to work with other people and try and solve some of these research questions together. So please do drop us an email. Some of the students we've had graduate from our lab are listed at the bottom, and very grateful for their work, and they've been fantastic to work with as well. And we have a small group of current PhD and Masters by Research students as well in our group. And this work is also supported by collaborations we have with some fantastic researchers at other institutions, and I'm incredibly grateful for being able to knock on their virtual door or in person sometimes and try and ask the questions that I need answers to, and they've been really, really helpful. So there's a really nice community of vascular researchers out there. I'm very grateful to having that support, and obviously, just like to acknowledge some of the grant funding that we've had to support our work, which has just been instrumental. Okay, so now that we've kind of, hopefully people are familiar with who we are and where we are, I suppose this slide sums up quite nicely why we started performing some vascular work in the first place. So this is about 7 or 8 years ago. I was doing my PhD here at Exeter, and I was very much interested in the primary prevention of cardiovascular disease. So overt, clinically relevant cardiovascular disease is obviously happening in later life, but it's preceded by this silent, slow, subclinical, progressive kind of covert progression of vascular changes. And typically we see changes in vascular function before we see changes in vascular structure. So I was very much interested in trying to understand vascular function early on, with primary prevention in mind. So sort of our work started off working in teenagers. So really thinking quite early for that primary prevention. What I was tasked in doing during my PhD was trying to, I suppose, develop our department's ability to perform a quite common vascular technique known as flow-mediated dilation, which I might shorten to FMD. For those of you who are familiar with the term, that'll be easy enough. But flow-mediated dilation is an ultrasound technique where you use a high-resolution linear array transducer probe on the brachial artery, and you're really trying to image the width of that artery. And what the technique involves is a blood pressure cuff, typically placed below the elbow, and there's some reasons why you might want to place the cuff in different positions, which I'm happy to talk about. And you measure the diameter of the artery, you inflate the cuff for 5 minutes, and that creates an ischemic challenge. Then you deflate that cuff for 5 minutes, and you have a hyperemic response. You have this increase in blood flow down to the hand. And what the ultrasonographer is trying to do there is trying to measure the response of that brachial artery. And in very simple terms, the wider the artery goes, you can express that as a change above baseline diameter, and the wider it goes, the better your endothelial function, speaking in generalities there. And studies have shown that this correlates with coronary artery function in the heart. So although we're measuring in the arm, it's sort of a non-invasive window into the heart. So think beyond the arm when this technique is being performed. But you can sort of see a screenshot there of this is us doing it in our lab. This is actually in a teenager. So the diameter of that brachial artery is quite small. It's 2.41 millimeters. And we have the pulse wave velocity at the bottom so we can understand blood flow. And really, we were using this vascular technique to try and understand vascular function. And we did some reliability work on that, and we kind of do our due diligence before we kind of rolled this out for research. But an early research study that we performed was trying to understand if one type of exercise is perhaps better than another type of exercise at acutely changing vascular function when measured through flow-mediated dilation. So just as a concept, I thought I'd share some of this early work from our group, just because I think it sets up the rest of the talk quite nicely. So On our y-axis, we have flow-mediated dilation, and the higher the number, the better, essentially. And we have teenagers coming into our lab. This is a within measures design, so they came in on 2 separate occasions, and they'd come in and we'd scan them, and then they'd perform some exercise, and they'd either perform high intensity interval exercise, that's the square, or the triangle for moderate intensity exercise. And what we can see is immediately after the exercise bout, after the high intensity interval exercise bout, flow mediated dilation is coming down. But then there's thought to be this biphasic response in vascular function after exercise. So we then tracked the response over time, and we can see that even after sort of 2 hours, high intensity interval exercise had improved vascular function. Now, we don't know the full time course of this response, but we've shown that here that different types of exercise can alter vascular function quite differently. And the important thing here is the amount of work that's performed in these 2 exercise bouts are the same. So if I was to describe that in lay terms, high intensity interval exercise is not better than moderate intensity exercise here because they've burnt more calories. They've done the same amount of work. So it's not quite the same when we think about calories and mechanical work. But I suppose the analogy here would be, is it better to jog for 100 calories or a sprint for 100 calories? And if one is better than the other, it's not because you burnt more calories. So we're trying to isolate exercise intensity as best we can. And here we've shown there is that independent effect of exercise intensity. So that was sort of quite interesting. And then at the end of my PhD, I came across this paper, which had actually been published in 2007 by Phil Ainsley. And they showed that, well, I suppose they were interested in understanding whether or not cerebrovascular function has a diurnal variation, a bit like peripheral vascular function as measured through FMD. So we know that FMD is lowest in the morning. At the time this paper was published, we knew that FMD was lowest in the morning. And we also knew that strokes tend to happen more frequently in the morning. And so the researchers had, on a very basic level, had a really interesting research question here of, well, if FMD is lowest in the morning, maybe cerebrovascular function is also lowest in the morning. So just to summarize why this is so important, these researchers measured cerebrovascular function and flow-mediated dilation, so peripheral vascular function, in the evening and then in the morning after a night's sleep. And what you can see on this figure here is that there was a correlation between the change in the overnight fall in vascular function in the arm and the overnight fall in vascular function in the brain. And if the change in something is correlated with the change in something else, it's quite a powerful way of suggesting that those things are associated. So when I think back to my own PhD work, here we've improved blood vessel function in the arm post high intensity interval exercise in teenagers. Perhaps we'd also improve blood vessel function in the brain. We just never measured it. So that was, the premise of our lab starting to do some cerebrovascular function. So really, we're interested in this link between cerebral and peripheral vascular function because we had done at this point lots of work looking at flow-mediated dilation. So our research questions quickly became, can exercise acutely alter cerebrovascular reactivity? And if so, is this intensity dependent? So and I'll pass over to Max now.
Cool. Thank you, Bert. And just to add one sort of extra level into the unique rationale for studying high intensity interval exercise and the brain, is this idea that the brain is a very unique organ. And with the periphery, we starting to suggest that the higher intensity, the higher the blood flow, the higher the shear stress, the better the acute and perhaps chronic adaptations to the exercise are. However, with the brain, we have to consider this almost risk versus reward approach, where we know that long-term exercise is good for the brain and it improves blood vessel function, and this helps reduce the risk of some cerebrovascular diseases. But there is this risk where perhaps with performing high-intensity interval exercise, particularly in some groups, we risk over-perfusing the brain with these large surges in blood pressure at the onset of exercise or the onset of those on intervals. And anybody that's done sprint interval training will certainly understand that's quite a big stimulus for the body to experience. And it's perhaps a juggling act of weighing up the risk of exercise, which may be more prominent in clinical groups or elderly individuals, versus the reward of the benefits of exercise. So for this study, we thought it sensible to start in healthy young adults and then hopefully set up a platform for which future research can build in more clinical or at-risk groups. So specifically off the back of this, the aim of our study was in young healthy adults to firstly investigate the effect of exercise intensity on peripheral vascular function using the technique flow-mediated dilation that Bert has already introduced. Secondly, we wanted to investigate the acute effect of exercise intensity on measures of cerebrovascular function, and I'll introduce the nuances of those approaches and how we utilize AD instruments to huge effect in that study. And then finally, we wanted to characterize the cerebral blood flow response during exercise as well as following exercise. As you know, sometimes to understand the benefits that exercise provides, it's really important to understand what is actually happening during the exercise and how does that differ between a continuous versus an interval exercise paradigm. So in terms of our methods, we recruited 10 healthy adults from the University of Exeter, and each adult visited the lab 5 times, performing exercise on a cycle ergometer on each visit. Visit 1 comprised of a ramp test. So for those that are unfamiliar, this involves on an exercise ergometer, in this case a bike, a 3-minute steady baseline period, and then a consistent increase in the work rate until volitional exhaustion. So typically in adults, that would be performed around 25 to 30 watts per minute. And the participant works as hard as they can, with the work rate getting progressively harder until they obtain VO2 max or VO2 peak. And we utilize that value to then prescribe 4 different exercise conditions that participants completed at least 2 days apart in a counterbalanced order to avoid any order effects between individuals. So on the next slide, I'm just going to focus on these 4 experimental conditions and how and why we designed the exercise trials as we did. So the first trial, so we've got the 4 exercise trials in different colors coming down the screen, and we've got time along the x-axis here lasting 30 minutes. So the first condition, our control trial, just comprised of 30 minutes of quiet seated rest in our laboratory. We then had MIE, which stands for moderate intensity exercise. And this was performed at 60% of each individual's VO2 max for 30 minutes, classically considered the moderate intensity protocol utilized in research. We then had 2 high intensity interval exercise bouts. 1 high intensity interval exercise 1 in blue and number 2 in red. And these comprised of a period of rest or recovery at 45% of VO2 max. And in high intensity interval exercise 1, we had 3 minutes of exercise performed at 75% of VO2 max. Interspersed by 2.5 minutes of recovery. And for high intensity interval exercise 2, we had 2 minutes on, 2 minutes off, but this time the on intervals were at a higher percentage of VO2 max, 90% as shown on the figure. And the reason that we chose these numbers was that during incremental exercise, cerebral blood flow is considered to peak in healthy adults around 75% of VO2 max. So we wanted to choose an exercise protocol where we think we might be hitting peak levels of blood flow, but also one of a higher intensity to, again, better understand the isolated effect of exercise intensity on these responses. And what's really neat about these exercise protocols is how we've managed to match them for a few important parameters. So the first is that we've time matched all these exercise conditions. So as you can see, hopefully, they all lasted 30 minutes. Okay, so if there are differences between trials, it's not because the exercise lasted longer in one condition compared to the other. And we were also able to manipulate for the high intensity interval protocols the amount of time spent on and the amount of time spent off to average out the average intensity. So as you can see on our figure here, all protocols, we targeted to average at 60% of VO2 max, but they all had an average percent VO2 max of 65. So this means that any differences that we see between the exercise conditions is not going to be an impact of differences in the duration of exercise or in the average intensity, which gives us more faith and confidence that any observed differences are due to the effect of the intensity of those bouts rather than any of these other important factors. And this was a real strength and something that we really, we had pens, papers, and whiteboards designing these protocols, and it was really good to see it come through. So in terms of how an experimental visit looked, participants arrived about 8 o'clock in the morning following an overnight fast, and we provided them with a cornflakes breakfast, which we know doesn't alter someone's vascular function. We then measured peripheral and cerebral vascular function before exercise and 1 and 3 hours following exercise. So in terms of how we specifically measured the different components of each of our aims, I've just repeated on this slide some information that Bert presented, illustrating how we can measure flow-mediated dilation using this ultrasound technique, where we occlude the forearm and basically measure the dilation of the brachial artery after a 5-minute occluded period. In terms of how we handled that data, I've just popped on the screen, we allometrically scaled that data. And what that means is that we can correct for any differences in baseline diameter between visits or within visits in each individual. So we analyzed those data following the latest guidelines available. And this is how we tackled our first aim of the study. So cerebral blood flow, I've just put a bit of information on this slide because I appreciate it's a novel and growing area. So apologies for anybody that knows this like the back of their hand. But this is the circle of Willis, the structure of the blood vessels inside the brain. And for us, the vessel of interest is the middle cerebral artery. So this is the largest of the intracranial blood vessels. And we can measure this using a technique called transcranial Doppler ultrasound. In terms of what it looks like in real life, here's a photo, I think from the first week of my PhD, so I've aged considerably. But we're able, once we insonate the artery, to pick up a trace that I've just popped in the corner of that image. And what we can see is that we are measuring the velocity of blood through that artery. And hopefully you can see that each time the heart beats, we get that characteristic Doppler profile of that velocity trace. And just one thing I wanted to quickly highlight is that what we are indeed measuring here is the velocity of blood inside the middle cerebral artery. So that's what MCAV stands for, the velocity of blood inside the middle cerebral artery. And just a quick point, we use this velocity measurement as a surrogate measure of blood flow, but obviously velocity and flow aren't the same thing. And they're only the same thing if we assume that the diameter of the vessel doesn't change. And this is something that's been hotly debated in the field, as I'm sure many of you are aware. But for the purposes of this talk and the wider literature, it's safe to say that it's a suitable surrogate measure of changes in blood flow if we use this technique to measure velocity. And the real advantages of this technique for our work, as you can see in the picture, we can attach the Doppler probes using a headset, which are fixed in place. So unlike the duplex ultrasound techniques where we need to hold things in place, we've got a headset and the participant is free to move. And that allows us to perform these measurements at rest, during exercise, which is how we tackled aim 3 of our study, and also during other whole body movements, such as squatting and standing, and there's many other things. The beauty is that you get excellent temporal resolution because it's fixed in place and the participant can perform this whole body upright exercise that with other techniques wouldn't necessarily be possible. Obviously, this study involved repeated visits to the lab, both within and between days. So just in terms of some important considerations that we need to address, it's really important that we replicate the scanning depth and position of the probe within an individual between visits. So there are often 3 different windows that you can scan on the sides of the head. So it's really important that you make a note of the position and the depth at which you're scanning, because obviously we want to minimize any confounding influence of that on the main findings. And just on the screen now, I've reported our coefficients of variation for baseline MCAV, both within and between day. And we've actually separately published our reliability data from this study in another paper, which I refer to slightly later on. So we were in our lab and we're starting to set up this technique. And one of the things that struck us very early on is that unlike other organs, the brain is very unique in that there's a left side and there's a right side. So for example, when measuring oxygen uptake, you can't measure the left lung separately to the right lung. But with this technique, you can measure the left and right side separately. So before we started the study, we dug into previous literature and we found that there was quite a lot of inconsistency in terms of how previous studies report the handling of bilateral data, or whether they use unilateral on one side or bilateral scanning. So actually, as part of my PhD, we wanted to address these inconsistencies. And we've got a study currently under review where we compared the left and the right MCAV responses to incremental and constant work rate exercise in healthy adults, and just came up with sort of a bullet point list of recommendations that we'd like to pass on to future researchers. And I've just put those on the screen now to share with you ahead of print. So we would recommend that if possible, researchers collect left and right sides to help continue to address these assumptions. And the added advantage of that approach is that the nature of this technique is sometimes participants may move their heads, their headset might slip slightly, you may lose a signal on one side. So the beauty of scanning bilaterally is that if you're only left with one signal, it's generally considered an appropriate surrogate measure of what's happening on both sides of the brain. We found that on a group level, unilateral measurement is okay, but we also found variations on an individual level. So the real need for that is if you're going to take a study where you have repeated measurements within an individual, so in our case, using an acute exercise model or in exercise training studies, for example, it's really important that you're consistent on the side in which you take that measurement so that you can minimize any confounding influences of left-right differences on any of your results. And it's also really important that future research continues to report how data are handled so we can start to add to a bit of consistency in the literature. So I just wanted to throw that as an early curveball we had in the lab where we're holding one probe and wondering which side to scan and why. So yeah, we've recently tackled that question. So That's how we measure cerebral blood velocity. How do we then translate a measure of velocity into a measure of function? So the brain is a very complex organ to study. It's receptive to many different changes and responds very quickly to many different stresses. So on the board is just the main 5 aspects that cerebral blood flow responds very quickly to. So these include metabolism, blood pressure. So specifically, the brain's ability to respond to changes in blood pressure is called cerebral autoregulation. So a nice lay example of cerebral autoregulation is if you ever stand up and you drop your blood pressure very quickly and you feel slightly lightheaded, that's your cerebral autoregulation failing you ever so slightly. So the idea is we want to maintain constant cerebral blood flow despite these fluctuations in blood pressure. It also responds to neurogenic and cardiac output changes, but the focus of our study was on what we consider cerebrovascular reactivity to arterial carbon dioxide. And that's how we tackled AIM2. So our measure for this study of cerebrovascular function was cerebrovascular reactivity to carbon dioxide. But this graph here shows that if we increase arterial CO2, we're looking at the red line on this figure, we increase cerebral blood flow. So our brain doesn't like carbon dioxide. If we're in a condition where we're elevating CO2, which is known to have an impact on the pH of the blood, cerebral blood flow increases as a protective mechanism to sort of wash out the brain. So there are many ways that we can induce what we call hypercapnia, where we increase the concentration of arterial CO2. One of the most straightforward is simply holding your breath, and CO2 obviously builds up during apnea. A scientific version of rebreathing that doesn't involve a paper bag. Or you can hook someone up with a mouthpiece or a face mask in a Douglas bag and actually get them to inhale air with a greater percentage of carbon dioxide than normal. However, our brain is also responsive to reductions in end-tidal CO2 or arterial CO2. And if we reduce end-tidal CO2, we reduce cerebral blood flow. An example of how we can induce hypocapnia to measure CVR in our labs is through different hyperventilation protocols. So for our study, we focused on one measure of hypercapnia, which was CO2 breathing. And we followed standard protocols at the time that administered 6% carbon dioxide for 4 minutes for our hypercapnic challenge. And for our hypocapnic challenge, we did 1 minute of hyperventilation at 25 breaths per minute. In terms of how these challenges then lead to a CVR outcome, well, we can measure the change in cerebral blood velocity during these challenges. We can measure the change in end-tidal CO2, which is our surrogate measure of arterial CO2, express them relative to each other, and that's our measurement of CVR. And similarly to FMD, the bigger the number, the healthier we consider, or the better we consider that response. So we want our brain to have as big a response to alterations in CO2 as we can. And as I mentioned earlier, we separately published within and between day reliability of how we handle CVR data using traditional and novel analytical approaches in experimental physiology about a year ago. And I hope that paper's been included in some sort of handout for you, but feel free to reach out to us if you'd like a full text of that paper. So in terms of what this looked like in the lab, this is an example, again, a younger face from me on the screen, but this is an example of what it looks like during exercise, and a number of our CVR assessments were performed in a supine position on the physio bed that you can see in the background. So we had our transcranial Doppler measuring bilateral or unilateral cerebral blood velocity. We could measure fingertip blood pressure using fingertip plethysmography, and we could also measure breath-by-breath changes in ventilation, expired gas, and in particular, end-tidal CO2, which is how we estimate changes in arterial CO2. We've got all these complex measurements going on, and this is where we really couldn't have performed this research without the assistance of AD instruments. So this is a power lab, and this can take 16 different inputs. So I've just put on the screen an example of how we can channel many different software, many different pieces of equipment, and time align these for the purposes of our research. So these include ECG, transcranial Doppler on left and right sides. This is a gas analyzer where we can measure O2 and CO2 changes. We can also plug in a spirometer to look at changes in volume, and our fingertip blood pressure monitor, which gives us beat-to-beat changes in finger pressure, mean arterial pressure, systolic and diastolic pressure, and also estimates of cardiac output, among other things. And the beauty of this is that this single box can run through a software that we call LabChart, and it allows us to have time-aligned data of all our different physiological responses in real time. So just an example of an output from one of our CO2 breathing files. What we've got in each row in each different color is a different physiological measurement. So you can see we've got our raw MCAV trace at the top, CO2s and O2s, pressures at the bottom. And we can also put in markers. So this shows the start of CO2 breathing. This shows the end of CO2 breathing. And what LabChart can also do is auto-calculate live in real time mean values. So we can derive MCAV mean beat to beat from the raw trace. We can calculate end-tidal CO2 from our raw CO2 trace. And the beauty of this is that we've got all our channels time aligned in real time, and all of our data can be exported together, which Bert would agree for the handling of these data is a huge advantage, and it saves you having to separately sync up different machines. So that's our methodology. We used repeated measures ANOVAs to handle most of our data. So in terms of what we found, our results, this graph here, very similar to what Bert presented in the introduction, is our FMD data. So on the y-axis, we've got our peripheral vascular function, and on the x-axis, we have time. So in the orange, our control trial, in the green, our moderate intensity trial. And again, in line with previous research, we've been unable to alter peripheral vascular function following moderate intensity exercise in healthy adults. Added to the graph now are both our high intensity interval trials, so at 75% in blue and at 90% in red. And we extend previous findings, and we show that the positive effect of high intensity interval exercise on FMD extends not just 1 hour, but also 3 hours post-exercise. So both HIIE-1 and HIIE-2 acutely improved FMD above baseline 1 and 3 hours following exercise. And we found no difference between the two HIIT conditions performed at 75 or 90% of VO2 max. So this provides further support that the greater the intensity of exercise, the greater the shear stress experienced by our blood vessels, the greater the acute changes in FMD. We were, however, unable to identify differences between 75% and 90% VO2 max, which may suggest that once exercise is of a sufficient intensity, the alterations in healthy adults at least will be the same, or that might be a consequence of how we averaged out the bouts between different intensities and durations. There's also some evidence that we help contribute to that the interval pattern nature of exercise may also be contributing to these changes in flow-mediated dilation post-exercise. Rather than providing a continuous stimulus, it appears that having periods of on and off where we experience those sharp changes in shear stress are positive for acute changes in vascular function. So for the first time, we documented changes in CVR to hyper and hypocapnia following these HIIT protocols. And on the left is our hypercapnic data, with the 4 trials shown in the different colors, and on the right, our hypocapnic response. And as is rather clear on the screen, we saw no changes in CVR following any of the exercise conditions at any time point in healthy young adults. This is in contrast to some previous work that found that after high intensity interval exercise, CVR actually dropped. And this suggests that, as I nodded to on previous slides, the different protocols of high intensity interval exercise and the different and complex measurement techniques to assess CVR may be underpinning these differences in previous work. And really, we're just sort of a few studies into fully understanding this field. Also, interestingly and excitingly, these data were in contrast with our peripheral vascular responses, which may be due to differences in vessel characteristics or different mechanisms shared between acute changes in the peripheral and cerebral vasculature. So in terms of vessel characteristics, we know that the resting shear of the cerebral arteries is about 4-fold that of the peripheral arteries. And maybe that perhaps is already at baseline, it's so high that at least in healthy adults, we can't acutely improve it any further. And that's something that perhaps excites me to continue this work in at-risk or clinical groups where perhaps the baseline levels are lower. Is there a scope for these acute exercise bouts to then lead to positive benefits? This also led us to scrutinize the MCAV responses during exercise because we saw no differences following exercise. What were the different responses of cerebral blood velocity during those exercise conditions? So this is our last slide of our results. And this time we've got time on the x-axis, but it's not in hours, it's in minutes. So this is during the exercise conditions. And on the y-axis, we have our velocity of the MCA. So our control condition, a nice steady line around 75 centimeters per second. This was our moderate condition, so 30 minutes at 60% of VO2 max. We see an initial rise in cerebral blood velocity and then a progressive decline towards exhaustion. This was our HIIE-1 protocol, and at the bottom of the figure, you can see the timing of the on and off bouts of exercise. And as the on intervals happen, we can see that we have an increase in cerebral blood velocity that decreases in magnitude with each subsequent bout. And our high intensity interval 2 protocol shown here in red. So our first finding of these was, well, the exercise conditions delivered very different stimuli and the pattern of MCAV differed between trials. But when it came to trying to put numbers and analyzing the differences between trials, we saw no difference in the 3 exercise MCAV responses when it came to the mean MCAV, the peak MCAV in each trial, or the area under curve to try and capture that whole stimulus across the 30-minute period. So perhaps the fact that we haven't been able to provide a different MCAV stimulus during exercise may be contributing to the lack of difference observed in CVR following exercise. And again, this is something that we've performed in healthy young adults where perhaps their baseline level of function is high enough that it can't be further improved. So again, this opens up many further questions for older populations, clinical populations, but also training studies where, okay, this doesn't have an acute response as a single bout, but what happens if you experience these changes multiple times across weeks, months, and training programs? So that's just an example of future research that we're looking to tackle in the future. So I've put a QR code up for the paper for those that haven't been able to access it, just while I quickly summarize the main take-home messages from our research. So we found that high-intensity interval exercise performed at both 75 and 90% of VO2 max improved peripheral vascular function 1 and 3 hours following exercise in healthy young adults. And this suggests perhaps a benefit of the greater brachial artery shear stress and or the interval pattern of exercise in these conditions. However, we found that CVR, or our measures of CVR, were unchanged following all of our exercise conditions at all time points, which suggests that perhaps changes that we observe in the peripheral vasculature aren't necessarily mirrored by changes in the cerebral vasculature. And this is the first study to highlight that the differential responses of peripheral versus cerebral vascular responses following acute exercise. And we highlight some potentially important influences of exercise dose. So the beauty of high intensity interval exercise is how we can manipulate the intensity and the duration of those bouts and the bout as a whole. Differences in measurement technique, so the vessels scanned, the challenges provided and how they're expressed, and also the timing of those. Are we missing changes? Are the changes yet to happen, for example? And also, as I've mentioned, perhaps differences in the sample may have different responses to the healthy young adults that we investigated in our study. So as I've mentioned, this research question certainly provided some answers, but certainly a lot more questions for our lab. So I'm just going to hand back to Bert now, who's going to summarize some of our current work that we're doing in our laboratory.
Yeah, sure. Thanks, Max. So I suppose that's a whirlwind tour of some of the work that we have performed. In truth, that work was funded by the Physiological Society from a grant that's now actually quite dated. So we got the money to do that work, and then with COVID disruption and some other things, it meant that actually took quite a long time for us to get that work done. And during that time, we had sort of set up other areas that we were interested to pursue. And we were also sort of developing our own ability to measure cerebrovascular function, if you like, a variety of different ways. And on the screen here, you can see a variety of different questions, I suppose, or exposures we're interested in. But from a methodological perspective, we're also, quite aware that breathing in a set concentration of carbon dioxide from a Douglas bag, i.e. in the study that we just presented, is sort of slightly dated now as a method for cerebrovascular activity or for CO2 loading. There's been some really nice papers in the last sort of 18 months or so. I think about Claire Burley or Jay Carr's work, which have sort of challenged cerebrovascular activity being assessed by breathing in gross amounts of carbon dioxide from a Douglas bag, for example. So one of the things we now have in our lab is we have a system where we can do end-tidal clamping of carbon dioxide. So we have a Respirac system, which we're really excited to sort of switch on for the first time in the next couple of weeks. So that's something that we're looking to do, and that will certainly improve our ability to answer these kind of questions. But we are looking at other cerebrovascular indices of health, if you like. So Max already mentioned autoregulation, so the defense of brain blood flow despite oscillations in blood pressure. So we are very much looking at that, and we're trying to understand how that might change in different groups or following different exposures. So we've just published quite a nice paper looking at resistance-trained individuals as well, because they are habitually exposed to changes in blood pressure as part of their training. We are also developing our ability to perform neurovascular coupling. So I know John Smurl has a neat methods paper looking at a Where's Wally task as a metabolic challenge. So we've done some work in that space, and that's been quite exciting. And we're applying that to, again, lots of different lifestyle risk factors as another indice of cerebrovascular reactivity. And then again, I think in addition to autoregulation, CO2 reactivity, and neurovascular coupling, I think we're both very interested in the responses during exercise, given that exercise is quite a profound challenge, both metabolic and then also dealing with changes in blood pressure and arterial carbon dioxide. So there's an awful lot going on there, and I think that exercise is a really neat, holistic challenge to try and understand brain blood flow regulation from a health perspective in different groups. And that could even be in healthy groups, but perhaps after a head impact or a concussion, for example. So that's some work that we're also doing here with some of my colleagues. So again, this is all supported with some really great typically young students who come in to our lab at postgraduate level. So that's been a privilege to be part of that as well. So that's very much some of the work that we're doing in this area. But I think the next step that we're probably both most excited by is getting the Respiract online, so we can do some end tidal clamping and perhaps address some of our initial research questions perhaps better than we could at the time we got the grant funding. So this field is certainly moving at pace. But I think that's all the things we kind of really wanted to cover with regards to the vascular techniques. As I said at the top of the talk, please do email myself and Max directly if you have questions. I know we're going to turn over to some Q&A now, and we can hopefully provide some answers to those questions. But if you'd like any more detail, or you're not comfortable raising a question in the chat, then you can of course just email us directly. So I suppose we stop sharing slides now, and Sydney will have some questions. I can see questions coming up actually.
Yeah, good.
Right then.
I think Sydney will read them.
Sure.
Thank you both so much for that presentation. Okay, are we ready to get started?
Absolutely.
Yeah, sure. So
fantastic. Our first question from Tina is wondering if all of the studies were conducted in men and women.
Sure. So thanks for the question. Sex differences is something that we're really interested in doing that work here. I don't know if we've ever been truly powered to be able to detect sex differences, but we've certainly recruited males and females for, I think, all of the studies that we do. I don't think there's been a single study, apart from some work that we're doing in women rugby players at the moment, and we've done some UEFA-funded work in women soccer players, looking at heading in soccer and cerebral autoregulation and neurovascular coupling. But I don't think we've ever excluded based upon sex, apart from when we've been looking at specifically sort of female athletes. But I don't think we were powered to detect an effect of sex, but certainly there were men and women, and I can't think of any study where it was men only.
And I think it's, especially in a growing field, is something that we'd recommend future research to continue to consider. And early research suggests that maybe in this healthy adult window, there's less of a difference, but different rates of puberty as children develop from childhood through to adolescence is going to have an impact, and also into later age. Some of the work that you've been a part of, Bert, has shown that there's differential effects in and around menopause, and the sex-specific effects perhaps happen later in life as well. So certainly something that future research needs to continue to tackle and better understand.
Thank you. I'd be remiss if I didn't mention Jodie Coop's PhD work, which was great to be involved in, and she demonstrated that there are changes post-menopause in neurovascular coupling that may have been missed in terms of that effect of the interaction of age and sex, because her data, she had some quite older adults, which was really nice to see age over the full life course to show those sex effects. So certainly very interested in exploring the effect of sex.
Fantastic. Thank you. Moving on to our next question, Thomas is wondering, when thinking about exercise dosage, what are your thoughts on using ratings of perceived exertion instead of percent of VO2?
do you mind if I jump
Okay.
in? Yeah, okay. So, just grab the mic. So we've done, well, some of my colleagues have done some of that work, looking at using rating of perceived exertion, mostly in teenagers. So from a pediatric perspective. My understanding is that rating of perceived exertion tallies quite closely to VO2 max when considered on a sliding scale. And, you know, it just has real ecological validity. So if you're prescribing someone an exercise intensity and you said 90% VO2 max, that may not translate particularly well to the individual, and it may not translate well to how you would be able to prescribe that in your lab if you don't have, you know, for example, a metabolic cart. So typically, when we describe an exercise stimulus, we'll actually revert to ratings of perceived exertion to help the participant understand, typically when they're giving informed consent. So yeah, I think rating of perceived exertion has a real place in prescribing exercise, certainly of a high intensity. And if I could just link that question to something we had on email in the week in advance of the talk, we had a really interesting email exchange with someone who was interested in high intensity interval exercise and the safety around high intensity interval exercise. And I think we're probably as a group quite keen to point out that high intensity interval exercise doesn't necessarily mean sprint interval exercise. It doesn't need to be boot camp, military training, throwing up in the corner type exercise, because that probably doesn't have much of a place in, you know, from a public health perspective. That's probably unachievable for many of us on a regular basis. So I think rating of perceived exertion would certainly be very helpful. I don't know if we reported rating of perceived exertion in our study. I don't think we did. But I would say that typically at the end of the high intensity interval exercise, if you're using a Borg 6 to 20 scale, people are around about sort of 18 and above towards the end of those, the high intensity interval exercise. So hopefully that gives you some context.
Thank you so much. That was a great answer. Our next question is curious about the role cortisol plays in these responses. Have you looked into that?
We haven't. We haven't really done much blood work. That's something that we'd be really interested to do. I think certainly with the study that Max presented, there was an awful lot of challenges to doing vascular research. I mean, so if you're I'm hoping that people watching this, and if you're a vascular researcher, heads are nodding right now when I say, you know, these things are, you know, really challenging to do. So sometimes when designing a study, I know I'm certainly guilty of trying to, to put too many. You don't have to agree with that. Of putting too many things in, but we didn't have the money to look at cortisol, so it's not something we've looked at directly. But you know, understanding some of those you know, blood markers would have been great. Yeah. I think we did initially try to look at things like BDNF from a cerebrovascular perspective, but I think, you know, our funding couldn't stretch that far.
It's always something to add on and do in the future, right?
Yeah.
That's great. Thank you. Do you think that this acute improvement in peripheral vascular function post-HIIT would be present the day after? So do you have any data for chronic effects of the HIIT training?
So that's a great question. Yeah, really exciting question, and that's one of the challenges. So there is some data back in, I'm going to say 2012, a paper got published which looked at performing exercise, high intensity interval exercise and moderate. I think the lead author was Tildum, and they actually included a measure of vascular function the following morning and then provided a postprandial challenge. And from memory, they demonstrated that the benefit post high intensity interval exercise was still there. So the length of the time course of benefit, that's a really good question. It's very challenging to do because at some point your participants are going to want to eat, and you know that can influence vascular function, and then they sleep, and we know sleep can influence vascular function. Then you have a circadian rhythm. So there's quite a burden to a participant in those sort of studies. It's certainly a study that I've planned on the whiteboard behind me, but never yet done. So I know that the benefits of high intensity interval exercise from our own lab team seems to extend to about 3, maybe 4 hours afterwards. We've never looked past that. We have, however, done a 2-week training study which looked at the benefit of high intensity interval exercise over that 2-week period. and we had a measure, the morning after the last exercise bout, and we showed there was a benefit there. And then we did 2 days of detraining, if you like, and then measured it again, and it was lost. And I know that a colleague of ours, Sasha Kreinen, his PhD, also had a 2-week detraining, but that was in adolescence. So I think there's perhaps no single study that I'm aware of that's answered that question. Certainly we haven't done that, but I think there is some data out there that you could probably infer if it's there the next day. But yeah, a great question.
Okay, yeah. Like before, there's lots of things that we can add on to do, right? New factors influencing all these things too. Okay, we have a question asking about what role you think SNS versus CO2 levels play in the CVF responses. So generally during moderate exercise, CO2 levels only moderately increases in CO2, but during HIIT training. Bit of a confusing question, hopefully.
Yeah. So I think what we're trying to understand there is what regulates the cerebral blood flow response to exercise. So apologies, John, if I've misinterpreted that question, just ask it again. But that's actually one of the focuses of my PhD, is how that's altered by age and maturation. And I think if you look at the HIIT protocol, we see these sort of cycles up and down of cerebral blood flow. And if we think of CO2 as you begin to hyperventilate, that's one of the main factors that drives the MCAV response down during exercise. So yeah, in adults, there's quite a strong body of evidence suggesting that CO2 is the primary regulator of those intensity dependent changes. But as always with the brain, there's so many other things going on. There's no perfect study where you can control for everything, but generally across sort of incremental, constant work rate and interval exercise, in adults, CO2 appears to be the primary regulator.
Fantastic. Thank you very much. Okay, let's move on to this next question. I think, okay, just moved up on my list. Cynthia's question: in your study, you measured CVR and FMD following different intensity exercise training. Did you measure carotid hemodynamics, for example, the pulsatility index? And then another part of this question: would differences in intensity influence the diameter of the MCA?
Do you want me to jump in, or do you want to have a go?
You can go.
Yeah. So one of the things when I mentioned some of when we didn't measure some blood markers like cortisol, there were other things that we wanted to do, and we ran out of. I ran out of talent. Basically, that's what happened. So we really wanted to try and get some understanding of what's happening at the carotid and pulsatility. Obviously, we can do some pulsatility index work, and Max is included in that, some of his PhD during exercise, for example. We are now just about to start a study which is going to be looking at, and this links into, I think, something else in the chat, arterial blood pressure changes as well, post high intensity interval exercise. And we're going to be, I think we're starting that study on Friday, actually. Yeah. So we're really excited to be doing that. So that's really, once we'd finished that work, that was a question that we'd really earmarked for future. So I'm really pleased to say that, we're about to start that work. I noticed that a lot of my answers to these questions seem to be, we're about to do
it's underway.
But that is truth, but that is truthfully happening. So listen, you're more than welcome to pop over, and if you're in the UK, if that's convenient, and see that in action, and maybe we could try and resolve some of those questions together, because it's challenging work, and we like having a very open door policy to the lab. So watch this space, but don't even watch this space. Don't be passive. Drop us an email, and we can try and include you in some of the work that we're doing, and that might give you perhaps a better answer than I'm giving you now.
Yes, that's fantastic. Really appreciate your openness. We did have a question from somebody asking about anyone doing this type of research preclinically. So that's an example. I think I would say reach out directly to you guys. Perhaps you have some information on who is doing that. And I think that would be the easiest way to answer that question. We are reaching the top of the hour. I think we can squeeze at least one more question in. So let's try and address Florence's question. The beneficial effect of HIIE on FMD is it a matter of high intensity or interval exercise?
Great question. Probably both. Again, we've done some work in Exeter in adolescents, which used the same sort of work matching paradigm, but the intensity of the intervals were moderate, and they did a number of repetitions to match the work done in high intensity interval exercise. And they found that there were the same or similar acute improvements in FMD in the moderate interval. So it appears that sort of the worst option is doing nothing. The next worst option is doing moderate continuous. And then if you introduce intensity or an interval pattern to your exercise, then those are the key mechanisms for the changes in shear stress during exercise that lead to those post-exercise benefits. Anything to add?
We know that the interval is also important from a a metabolic and even cell signaling perspective. There's been some work done on that out of UBC. I think part of our work where we compare high intensity interval exercise to moderate intensity continuous exercise, that there's a limitation there. I mean, mathematically, it helped us to get a 30-minute stimulus to average 60% VO2 max, but we perhaps could have included some recovery intervals, if you like, in the moderate, and that would have been of interest. So I think the interval nature of exercise could be quite important, but we'll have to see.
Fantastic. Thank you so much. So we have reached the top of the hour. We still have so many questions. So, everybody continue to submit them. I am going to send them off to Bert and Max, and they'll be so kind as to take the time to try and provide some written responses to these. So please continue to submit your questions. But with that, I do want to say thank you so much to our wonderful speakers. It was such a pleasure to meet you and hear about your research and to have you here with us today. So I hope you enjoyed yourselves.
Thank you. Thanks for organizing, Sydney.
Thank you so much, guys, for listening, whatever time it is for you as well.
In closing, we hope you enjoyed this Inside Scientific webinar, produced in partnership with the American Physiological Society and sponsored by 80 Instruments, and I hope that you enjoyed the rest of your day.